Okay, it's Dr. Goldsmith again. This is the fourth in the series of videos that I'm recording in my absence. So far we have actually seen the antigen presenting cell present antigen to the T cell, so had antigen recognition occur, but that alone is not enough for expansion and differentiation. There have to be some other signals that go on in addition to what was just described. So I'll continue talking about the various signals that we get to get the full expansion and differentiation of the T cells. So I'm going to describe over the next few minutes survival and clonal expansion, so promotion or inhibition of T cell survival and expansion, that is clonal proliferation. This really, there's a key molecule involved here, that's B7. It's a member of the immunoglobulin superfamily, and it's found exclusively on surfaces of cell that stimulate naive T cell proliferation. I will also talk about the differentiation process to ultimately become effector cells. All right, so we need some co-stimulators for full activation of the T cell. It's not enough just to have the antigen interacting and the signaling through CD3 and zeta and so on. We get these co-stimulators. They're provided by the antigen presenting cells, and the most well known are B7-1 and B7-2. It's enough to just call them B7. That's sufficient, and that is what I will be using throughout. It is a family of proteins or co-stimulators, but that's enough for you to know. You don't need to know more than that. They are expressed by the antigen presenting cells, and their expression is increased when the antigen presenting cells meet microbes. So the beautiful thing here is, when you're talking about having those second signals, we're talking about antigen presenting cells that have met microbes are going to produce things like B7 that will help to fully activate the naive mature T cells. B7 is actually recognized by CD28, which is a receptor expressed on almost all T cells, and it's the CD28 mediated signaling that's essential for responses of these naive T cells. So it works in concert with signals generated by the binding of the T cell receptor and its co-receptor to the peptide MHC complexes. This will give you the full activation of the naive mature B cell. There is another helpful molecule for T cell co-stimulation, and that is CD40. This is really an indirect method of helping with co-stimulation, and what do I mean by that? We find CD40 on antigen presenting cells, and this in turn increases co-stimulatory signals for T cell responses by making the antigen presenting cells better stimulators. The CD40 on the antigen presenting cell binds to CD40 ligand, which is also known as CD154. CD40 ligand is sufficient. That's found on the T cells, and CD40 ligand is expressed on antigen-stimulated T cells and binds to CD40 on the antigen-presenting cell. I say it's indirect method of co-stimulation because this is going to cause the antigen-presenting cell to produce and express more B7 co-stimulator, which you just heard about. In addition, the antigen-presenting cell will secrete cytokines like IL-12, and this is going to enhance the T cell differentiation into fully activated T cells. So it's an indirect method of getting more B7. The interesting thing about this is the co-stimulation that's necessary for full T cell activation really does explain an observation that protein antigens alone, like those found in vaccines, for example, won't elicit a T cell dependent immune response unless they're administered with other substances that activate antigen presenting cells. So when a vaccine is made, and if it's to a peptide antigen, there are other substances known as adjuvants, ADJUVANTS. These adjuvants function mainly to induce expression of the co-stimulatory molecules on the antigen presenting cells. So it's kind of cool use of the fact that we know that co-stimulation really helps to rev up and fully activate the naive T cells. A very common adjuvant is alum. It's an aluminum gel or salt, and it's really in vaccines for eliciting T cell responses. So here's this illustrated. We have our antigen recognition. We have the antigen presenting cell with an MHC molecule and a peptide, and it's being recognized by the naive T cell, the T cell receptor. They're not showing all the co-receptor. They're not showing the signaling molecules either. What they're really focused on is looking at the accessory molecules for co-stimulation. So you can see CD28 is on the T cell, but it's not engaged, and so you get no response to this antigen recognition. In contrast, to fully activate the T cell, if the antigen presenting cell is activated by microbes or other things from the innate immune response, then you will get this expression of B7, increased expression of B7, which will bind to the CD28 molecule on the T cell. This in turn will allow for secretion of cytokines, and the T cell will be able to proliferate, becomes fully activated, it will proliferate and differentiate. So that's very cool, and it tells you, hey, yep, we do need more than just antigen recognition for signaling to be appropriate and for the T cell to be activated. Now, if you can stimulate a response, how do you terminate a response? Well, you do that through the same molecule, through the CD28 on the T cell. It's actually really, really cool. There are proteins that are homologous to CD28, and they limit or terminate the immune responses. So there are different members of the CD28 family, and they participate, different members, in activation or inhibition. Inhibitory receptors that are members of this family include CTLA4 and PD1. And you'll note, if you go back to the earlier table that I showed you and said, oh, this is going to come in handy, you will find them on it. CTLA4 recognizes B7 on antigen presenting cells, just as CD28 did, but it is inhibitory. PD21 recognizes a lot of different related ligands on multiple cell types, and both CTLA4 and PD1 are induced on activated T cells. So really what this boils down to is you get sort of a seesaw, if you will, or a balancing act between activation signals and inhibition signals. There are questions that remain. How do the inhibitory pathways become active? What's the choice between activation and inhibition? How is that determined? It's not entirely clear. But I can tell you it is useful. So this is an application that I think some of you may actually have heard of this. Orencia, also known as abatacept, it's a monoclonal antibody. It's an immunotherapeutic agent used to treat rheumatoid arthritis, which is an inflammatory disorder. And it's an autoimmune disease. It is believed that T cell activation is implicated in the pathogenesis of rheumatoid arthritis, and so what has been discovered to work is a fusion protein of this CTLA4, so its extracellular domain fused to the Fc region of an immunoglobulin molecule. Now, if you treat someone with this fusion molecule, what would it mean in terms of T cell activation? So I'll let you think about it. It'll be activated, T cells will be inhibited, T cells will proliferate, T cells will secrete cytokines. Sorry, my check mark didn't show up. But since CTLA4 is an inhibitory molecule, the T cell activation would be inhibited. Now, there's one final thing to think about, and that is, what are the stimuli that are necessary for T cell activation? I mentioned this before. It's slightly different than for CD positive T cell activation, CD4 positive T cell activation, in that we know that activation requires MHC class 1 plus peptide and co-stimulation. It also appears that CD8 positive, naive, never seen their antigen before, now they're seeing it for the first time, naive, mature CD8 positive T cells require the help of T cells to be fully activated. So again, this is slightly different from other T cells. It looks like they take the cytoplasmic antigen from one cell that's being cross-presented by dendritic cells for the initiation of the CD8, mature but naive CD8 T cell to be activated. And they then will be able to differentiate into cytotoxic T cells. It does appear that it requires concomitant activation of CD4 positive helper T cells. I've noted this before. Here this is again in picture form, where we have this cross-presentation. We have an antigen presenting cell presenting antigen on processed antigen on MHC class 1 plus the co-stimulators that were just described to the naive but mature CD8 positive T cell. It does appear that these naive CD8 positive T cells may require the help of T cells that have been activated, and to essentially the same microbe can then stimulate the proliferation and differentiation of the CD8 positive T cell. So once the T cell is activated, what happens? Well, not surprisingly, you get a lot of signaling and biochemical pathways that are activated upon T cell activation. So I'm going to talk about those for a few minutes, and then I will stop recording and we will take a little break, at least for me, and then I'll record the last part of this. So biochemical pathways that are activated when the T cell is fully activated, not surprisingly, you get protein expression when T cells recognize antigen plus their co-stimulators. Again, we're talking mature naive T cells that are seeing their antigens and their co-stimulators for the first time. The proteins that are expressed, not surprisingly, are involved in proliferation, differentiation, and effector function. Just what you would expect if you want to fully activate and expand the population that's responding to a microbe. The antigen recognition linked to the T cell responses is done so by proteins. There are enzymes, adapter proteins, transcription factors, things that you associate with second messenger systems as a result of engaging receptors on the cell surface of a cell, in this case, the T cell. So again, let me remind you that these types of proteins would be conserved. They don't need to bind antigen, so they don't need to be diverse, but there are lots of them. So what are we going to see? Well, it takes a little while, which is why adaptive immunity takes some time, but over time you will see things like IL-2 expressed. I'm going to talk specifically about that. You'll see CD40 ligand expressed. I just mentioned that with regard to co-stimulation. You'll see an IL-2 receptor chain expressed to a greater degree than it was before. And ultimately, you're going to get proliferation and differentiation. All right, so what are these molecules? Well, you need to know that when the molecules are found together, they actually form a big synapse. It's called an immunologic or an immunological synapse. Essentially, this is a region of contact between the T cell and the antigen presenting cell. You have your T cell receptor complex that would include the signaling chains. You have CD4 and CD8 co-receptors, depending upon whether you have a CD4 positive or a CD8 positive cell. You have CD28 at the center of contact that's there for co-stimulation. And around that, there are integrins, adhesion molecules that form a ring. And all of these things coming together at this immunological synapse is believed to induce interactions that are required for optimal signaling within the T cell for activation. The inhibitory signaling molecules like CTLA4 and PD1 are also recruited to this area. And I have a nice video that I will show you.
In this 3D image, we are able to see the interaction between a T cell, colored blue, and presenting cell, in this case a B cell, colored red. When T cells are able to recognize their antigen on another cell, the zone of contact between the two cells, here colored green, forms an organized structure called the immunological synapse. In this second image, which shows a cytotoxic T cell recognizing its target, we can again see the synapse between the two cells. The cells are labeled with fluorescent molecules that show an adhesion molecule, the integrin alpha chain CD11A, in green, and a signaling molecule, LCK, in red. The cytotoxic granules in the CTL are labeled blue. If we look closely at the synapse, we can see its structure. The outer ring contains the adhesion molecule, and the inner ring is divided into two parts, one containing the signaling molecules, and the other, dark in this image, is the secretory zone. In this side view, we can see that some of the cytotoxic granules, stained in blue, have moved close to the interface and are starting to fuse with the synapse. Other granules remain at the opposite end of the cell, perhaps where another synapse is starting to form. Multiple synapses can be formed with the same antigen-presenting cell, as we see here, where two CTLs are trying to kill the same target. Each has formed a synapse organized into discrete signaling and secretory zones. The signaling zones are indicated by the presence of the red-stained signaling molecule, LCK, while green-labeled cytotoxic granules can be seen clustered just behind the synapse itself.
So that's pretty cool. The cytotoxic granules will come into play when the cell is an effector cell. Didn't mention it here, but the point of that is to show you that immunologic synapse with all of the various molecules that are found within that synapse. There was some mention of some of the signaling molecules like LIC. I'm going to spend a little time talking about them right now. I would like to refresh your memories that the T cell receptor complex is an alpha-beta dimer, a heterodimer. It's required for antigen binding, and it forms a complex with several signaling proteins like CD3 and zeta, things you already know. And CD3 is a 3 protein complex, and zeta is a heterodimer. Again, things you've seen before. The CD3 complex binds to non-covalently to the T cell receptor, and it allows the whole complex to be transported to the membrane. And again, there are multiple motifs, immunoreceptor tyrosine-based, in this case, activation motifs, that are found on these signaling molecule chains. They are there to allow for signaling. A lot of phosphorylation occurs on these, and they're highly conserved because they don't need to do anything but signal. So unlike the antigen binding area of the T cell receptor, the signaling molecules are highly conserved. The ITAMs themselves are found for B cell receptor signaling chains, NK cell receptor signaling chains. They're found on FC receptors of mast cells, of macrophages, monocytes, neutrophils, highly conserved motifs. They actually contain two serines, that's the Y, that can be phosphorylated by kinase. All right, so what happens? Well, a lot of phosphorylation, and more phosphorylation, and more phosphorylation. Essentially, engagement of the receptor plus those accessory signaling proteins plus the co-stimulators induces a cascade of phosphorylation. The first thing that happens is antigen binding leads to phosphorylation of the tyrosines on those motifs, on the ITAMs. And the B cells and the T cells actually follow similar signaling pathways to the nucleus, but they have some different signaling molecules, and certainly the outcomes of those events for protein transcription would be different. Ultimately, the signaling leads to the nucleus. This involves transcription factors being activated, and these induce gene transcription. So hopefully you remember back from the early videos that you were asked to look at on your own or read the information on your own about signaling. This is very standard signaling. Some of the kinases that are involved include LIC, I know it's a great name, LCK, and then some very conserved enzymes like phospholipase C gamma, PLC gamma, which catalyzes the breakdown of a membrane protein, phosphatidylinositol P2. So phospholipase C catalyzes the breakdown of that membrane lipid PIP, phospholipase inositol P2, and that produces a second messenger, inositol 1,4,5 triphosphate, or IP3. And there's also a membrane-bound lipid produced from this, stays at the membrane, and that is diacylglycerol, or DAG. Again, hopefully this is familiar to you from the second messenger information you were asked to do early on this semester. These signaling molecules have specific responses themselves. IP3 binds to the endoplasmic reticulum on specific receptors and releases calcium, which has other effects I'll show you in just a moment. Diacylglycerol stays at the membrane and activates kinases. So phosphorylation ensues. There are also small G proteins involved, like RAS, and these induce kinase cascades themselves. And there are transcription factors at the end that will cause gene transcriptions like NF-kappa B, NFAT, and AP1. So here are these molecules shown in diagram fashion, where we have our antigen-presenting cell at the top, our T cell here with the various molecules of interest. We have our T cell receptor that's meeting MHC plus antigen. We have our co-receptor, CD4 or CD8. We have our non-covalently bound signaling molecules, CD3 and zeta, with their ITAM motifs that will get phosphorylated on serines. We have the protein kinase LIC on the co-receptor that gets phosphorylation underway. We have some accessory proteins that dock and allow for further phosphorylation. We also have small G proteins that will exchange phosphate groups to become activated. And we have phospholipase C that is going to activate the membrane lipid PIP2. And ultimately, we get many, many signaling molecules as a result. So phospholipase C activation ultimately causes IP3 that binds to the endoplasmic reticulum and releases calcium from the endoplasmic reticulum. Diacylglycerol remains at the plasma membrane. These in turn activate other enzymes like PKC, phosphokinase C, or calcineurin in the case of calcium. If you're talking about GTP, GDP, small G protein exchanges like RAS and RAC, these become phosphorylated to be active. So we have our small active G proteins. They, in turn, induce the signaling for active enzymes ERK and JNK. And PIP3 also will allow for activation of various enzymes. I don't care so much that you know the names of these enzymes. That's okay. What I care about is that you know the initial steps: phosphorylation, activation of small G proteins, IP3, and phospholipase C, and IP3 being produced from PIP2, producing IP3 and DAG. And then I do hope that you'll remember some of these transcription factors like NFAT and NF-kappa B. So we see all of these many proteins at the plasma membrane at the site of that immunological synapse. It's incredible what goes on there, and it's highly, highly active. The result of the activation of these transcription factors, of course, is you're going to get protein synthesis, which is beautiful. And the proteins that will be synthesized will be involved in proliferation, differentiation, and ultimately effector function. So I have a question for you, and that is, T cell activation cannot be limited. Is this true or false? I'll let you think about that, and the answer is, it is false. Absolutely, you can limit T cell activation. You're going to do it through those inhibitory receptors, CTLA4 or PD1. They actually have motifs themselves. They're immunotyrosine inhibitory motifs, so they're ITIMs, not ITAMs. All right, I will stop here, and the next thing we will pick up will be with regard to cytokines.